You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Fedor Galkin. He's coming from Moscow. It's probably pretty late where he is. I appreciate him coming.
This is Fedor.
Yeah, Fedor, thank you for coming. How you doing?
Well, I'm doing great. And you?
Yeah, good. I wanted to talk to you about how you're correlating the age of someone by looking at their microbiome, human microbiome aging clocks.
Yes.
Tell me, what's your, where do you do this work through, and how did you get interested in this?
Well, first of all, I graduated from Moscow State University, Faculty of Bioengineering and Bioinformatics. And when I graduated, I entered this company called Insilico Medicine. It is based in Hong Kong, and it focuses on age-related studies, longevity science, aging biomarkers, and all such things. And also a lot of drug design, but we have a great lot of studies dedicated to longevity science. And we had this project to develop microbiome aging clocks. There are all sorts of aging clocks today. The first of them were developed in 2013 by Harvard and independently by Hannam et al. They developed DNA methylation aging clocks. This is a technology that lets you read specific kind of marks on your DNA called DNA methylation marks, and based on your profile, you can. This method lets you guess the person's age. So since then, there have been developed a lot of similar technologies that let people guess somebody else's age based on their blood biochemistry, based on their gene expression levels, and all such things. But there has never been such a clock that would take in the abundance of microbes that live in your gut and predict a person's age. There have been quite a few studies that show some correlations between the abundance of specific microbes and the age of the person. But these studies have never gone beyond proclaiming some correlations and deriving some kind of dynamic theory or developing a reliable predictor. And here we are. We have used neural networks to build our predictor. And it is quite, I don't want to say radical, but quite unusual in the field, because most of the aging clocks, they use much simpler algorithms. For example, linear regression, elastic net, or something like this, to take in the quantified biomarkers. Say if we deal with DNA methylation, we feed to such an elastic network, we feed the percentage of methylation on a subset of methylation sites, and it produces a linear model like y equals some coefficient times x plus b, something like that. And
well, okay. First of all, I mean,
yes,
there. How
sorry.
many different? No, it's all right. How many different possible markers can you look for, and how would you ever know that there's not dozens of cofactors? And it just seems really difficult to model any of this stuff. Like, how do you even approach it?
So there are different ways of approaching it, but we have gone for the most straightforward one. We have chosen the most abundant and the most prevalent in the population microbes. So we had a cutoff of, if I remember correctly, 5%. So each microbe our model uses is present in at least 5% of the population we studied, and it is present in each person. It is, well, not in each person. On average, it is present. Its abundance is no less than 10 to the power of -5. So we just had some thresholds to take only the most prevalent and abundant microbes.
So in all the samples that you've looked at, what kind of for the prevalent microbes, what kind of percentages do they comprise? Have you had situations where one microbe is like 20% of the entire flora of someone's gut? What's the range you've seen?
Well, we've seen quite abundant microbes that, as you just said, can take dozens of percents. And we have also taken the microbes that are rather minor occurrence in the gut, and the total set of microbes our model can use is above 1,000. But typically, we do not see more than, say, 200 microbes in any particular person.
Oh, you only see 200 microbes total, different ones?
Well, usually in each person, we see only 200 microbes or something like that.
But again, the most prevalent ones, what's the maximum percentage you've seen in somebody of any given species?
And that would be something like 50%, I guess. Well,
Really?
Bacteroides genus and the bacterium genus, Akkermansia, they are all quite high prevalence microbes.
But that's at the genus level. At the strain or species level.
Oh yes, I was talking about the species from these genus.
Oh, so you've seen a single species that represents up to even 50%
yes,
of someone's microbiome?
Yeah.
So how does that correlate? Yeah, tell me about some of the clocks and some of the correlations. So have you seen that if someone has one or two species that make up a very large percentage of the microbiome, does that typically mean they're unhealthy or not necessarily?
Well, not necessarily, because you have to inspect these people thoroughly to understand whether they are healthy or not. But we have trained our model only on people that were marked as healthy or control in some kind of study. Sure, we know that if a person is marked as control, it doesn't mean that he is completely healthy. But still, we hope he's healthier than somebody in the case group of a study.
Okay, and then in terms of the clocks, what have people found in the literature? What determines a person, or not determines, but what do they see at different ages in people? What changes in their microbiome?
So, speaking on that, there are some correlations. Well, the most widely known correlation is on very high level of organization. So there are two big groups of bacteria called Firmicutes and Bacteroides. And it is said that with age, the fraction of Bacteroides to Firmicutes, it fluctuates. So, say small children, they first have big fraction of Bacteroides to Firmicutes. Then in adult people, it actually decreases. And then the older a person gets, Bacteroides to Firmicutes ratio goes higher and higher. So that is one of correlations we can find in the literature. Also,
is it all right? Look, for that correlation, is it linear with age, or is it exponential, or what does it look like, that curve or that that change over time?
Well, I'd say it is something moreover, what do you call it, reversed bell or something like that.
Oh, so is it a step function as well? Or does it stay for a few years and then is it always changing, or does it stay stagnant or constant for a while and then suddenly change?
It is changing, but as I said, this fraction, it is high both in children and in elderly people. And
is it the same fraction, or does it change, like Firmicutes to F to B, Firmicutes to Bacteroidetes? Well, I'll just call it F and B. So what's the ratio in kids and what's the ratio in old people?
In old people, it is something like if we talk F to B, right? F to B, it would be like 1/10, and in adults it would be like 2 or 3 B to F, and in children we also see this something like 1/10.
So kids, we'll keep it constant as F over B, let's say. So it's 1/10 in kids, then it goes 3/10 in adults, and then back to 1/10 for elderly people.
Wait, no. If we talk B to F, it is something like 10.
Let's keep the same ratio. Yeah, let's keep the same ratio. You decide which one, but
all right. So
those three segments, what is the ratio?
B to F, it is something like 10, then 0.5, and then 10 again.
Oh wow, it changes. Okay, flip flops, and then goes back. I see what you mean.
So it is something like a reversed bell curve or something like that.
Like a U shape, let's say, over time. Okay. Interesting.
Yeah. So
So what does that tell you? Seeing that, what does that tell you to look for or to consider?
Well, we have not studied our model on such high levels. We jumped straight to the species level. And we found we have some findings that align with the literature on species, because, although this is a really general correlation about Bacteroides and Firmicutes, it is not suitable for accurate prediction.
Okay. What are you trying to predict, by the way? Someone's age or other factors?
Primarily someone's age. But we have also discovered that this model of ours works fine on people with normal microbiota. It predicts them accurately, but it fails completely on people with type 1 diabetes. So we assume that there is something wrong with the microbiota in people with diabetes, and this is somehow affecting the process of intestinal aging.
Type 1 or type 2?
Type 1. We have only tried type 1, no type 2 yet.
Oh, that's interesting. Type 2 would be, I think, really interesting, but type 1 is, I'm sure, too.
Yeah, we're also looking for other kinds of data to test with our model, to verify it further and to see what other diseases have such an effect on microflora that we can catch with our model. So if anybody out there is interested in cooperating in this direction, we would gladly discuss academic or even commercial partnership.
So for type 1 diabetes, what does it look like? Have you done a longitudinal look yet? Have you gotten people at various ages that have type 1?
We have people of various ages, no longitudinal yet. But what we see is that the younger people with type 1 diabetes, they are predicted to be much older than they actually are. So it seems like the people with diabetes, they constantly have their gut in an old stage. Either if a person is young or if the person is old, all we see is an old person.
Oh, wow. How old approximately?
Well, something like a 20-year-old guy with diabetes could look to this model as a 60-year-old guy.
Okay, interesting. And what about a 60-year-old guy? Do they look 80 or they still look 60?
He would probably look something like his age.
Okay.
So, yeah. So typically we see that younger people seem older, but older people still look old, as old as they actually seem to be.
Have you looked at how diet influences the this curve, even for healthy people?
We're trying to develop this direction right now, but it is not at all easy to deal with diets, because first you have to find some fine studies where this diet is well documented, and perfectly you would like to see some person in different time points. And also another limitation we have is that we deal only with whole genome sequencing data. So a lot of studies out there are made with 16S sequencing technology, and that limits our selection of data we can use. So
what's wrong with doesn't 16S only look at parts of bacteria that seem to be preserved over a long period of time? That's why they use it?
Yeah, well, they use it primarily because it is much cheaper than whole genome sequencing, and also data analysis, or say classification, is also goes much faster, much easier than WGS sequencing runs. So,
why can't you use why can't you use the 16S for your models? If you can't get whole genome sequencing and you want to go faster, why don't you use 16S as a proxy for that?
Well, first of all, we have tried doing some 16S, and we believe that the level of resolution we can obtain with 16S technology is not enough. So we went with WGS because then we can achieve much more accurate classification results and see the actual species. Because with 16S runs, it is quite often the case that you only can tell different genus or gene apart, not species. And
what is shotgun sequencing? I've heard about that. Is that the same as whole genome sequencing?
Yeah. It is the same. So if 16S sequencing is looking for a specific marker gene that can tell two different species apart, the whole genome or shotgun sequencing, it breaks up the whole genome of any bacteria, and what you receive is a genome of a bacteria breaking in lots of different ways in different parts. You get small pieces of text from this genome, and then you can be much more sure that this particular sample has say 10% of a bacterium from species A, because while breaking the genome of this species A in your sample, you get not only one marker gene, but you get like tens and hundreds of marker genes from all kinds of bacteria, and you get to classify your runs much more accurately.
Okay, gotcha. Well, why not? Why don't you? I mean, just as a directional type thing, why wouldn't you approach someone that says they're a carnivore, or someone that says they're a vegan, someone says that they just have like a standard diet, and look at like 2 age points, old and young, and just see what you see for a starting point, and maybe get a directional sense of what you could find if you went more in detail.
Well, that is one way to look at it, but first of all, we decided to see if we can simulate the effects of different diets. there is a lot of information in the literature on what kind of nutrients causes what species to uprise and downregulates the other species. So we decided to go with that information first and see, say, if we have a person and we know his age and his microflora profile, so the abundances of all bacteria living in his gut, and try to digitally feed him more protein or more glucose, and see what and simulate the upregulation glucose causes, and then predict the new, and then we predict the new simulated profile and see what happened to his age prediction, if it goes higher or lower. So unfortunately, so far we have not received any steady results on this. This may be the case with the literature data, because most frequently you cannot quantify these effect, say glucose causes, very accurately. You can only say if glucose downregulates or upregulates a microbe.
Okay.
So it is not accurate enough for us. So we should know what exactly how strong the effect of any nutrient. And it seems like nutrients don't act that independently from each other, and you need to take in the information on the whole diet. And
what do you mean nutrients don't act independently of each other? What do you mean?
Well, apart from a nutrient being consumed, it is quite important to consider the source of the nutrient. So you can say take supplements and get more, say, some kind of a vitamin say E or something like that, or you can take some kind of a product. You can take your vitamins with whole grain bread, with milk, with meat, vegetables, and what the nutrients come in tow with, that greatly influences the effect on the microbiome.
Okay. So, I mean, there's a lot of directions you can take this. Where do you want your focus to be, and why? Like, what do you want to figure out specifically?
Well, first of all, we would like to verify our model further, and if we need to, we would like to overtrain it. So we would like to add more data to let it consider all the variation in human microflora that possibly exists. So first and foremost, we would like to train our model on more data. Then we would like to see if we can pinpoint some diseases akin to type 1 diabetes with this clock, and possibly see the effects of medication, of diseases and medication together.
How variable are people's microbiomes? Healthy people, like,
well,
how many samples have you seen, and how different are they?
They're extremely variable. So altogether, we have seen actually over 4,000 profiles from 1,000 and something like 700 people. We saw that they're actually all quite different. For example, as I've said, we have a model that can take in something like 1,000 microbe abundances, but at the same time we observe only 200 or 300 different microbes in each person, and it is quite frequently that these different microbes overlap only slightly.
Mm-hmm.
You can take two different people and say one lives in China, the other lives in US, and they will have only say a 15% overlap of the total community abundance.
Well, what does that tell you? Does that tell you that there are various microbes that can do the same job apparently in people, in their hosts, and maybe that's
Well,
why there's such variation, yet still have health?
Well, yes, it is actually one of the other reasons we went for whole genome sequencing. It is widely known that there is no such thing as universal healthy core microbiota, something that all human people share. You can have a healthy microbiota in 1,000 different ways, but what is more or less stable is the functions this microbiota carries. It was found within the Human Microbiome Project. They studied the species composition of microbiota, and then they looked at functional composition of microbiota. They used whole genome sequencing to classify the genetic information, not in terms of species and taxonomy, but in terms of genes and what they do. And what you see when you look at human microbiome like this, you see that although two people can have absolutely different taxonomic compositions, they actually have quite similar function distribution in their microbiota. So although human microbiota is a very variable object, it still has something common in it. And another thing we wanted to see is what kind of a model we would get if we used genes contained in the gut instead of species. But so far we still haven't run out of things to do with on species level, and when we do, we will probably go down to functional and genetic level to see what is happening there.
Well, what about on the metabolomic level? What if you look at your 700 people or 1,000 people on the metabolomic level and look at the prevalence of various metabolites? Maybe then they'll look much more similar.
Yeah, probably that is what we expect, but we have not done that yet.
Okay. Yeah, I just wonder on some level there's going to be they'll look much more common than not. So that's just my speculation. But do you deal with
that is probably the case.
Where are your samples coming from? Is it oral? Is it fecal? Vaginal? Or are you doing fecal. Multiple sites?
No, we're doing only fecal samples, and all we use are the samples obtained from public repositories like European Nucleotide Archive. We found practically all our samples in there. So, and we were almost always looking for the samples that had metadata with them. So where each sample had the information on what kind of person it came from, like age, gender, if the person consumes antibiotics, if the person has some kind of a disease or something like that. So we use this kind of data.
Interesting. You have a lot of work to do. So what is going to be your focus over the next year or two? What do you want to figure out?
Well, we would like to figure out, the actual purpose of this whole project is to see if we can find the silver bullet microbes that can make people look younger or actually make them look older and effect on these microbes. So if we find, say, a geroprotective microbe, we want to make it flourish in our guts so that it makes us younger. And then we would like to possibly see if it can be administered as a probiotic to people, or if there are specific dietary interventions people could take to increase the abundance of these bacterium, or maybe lifestyle decisions like, I don't know, quit smoking or drink less, and the effects these interventions cause on your longevity are actually mediated by some kind of a geroprotective bacterium.
I guess another interesting thing is if you look at hundreds of people, and you look at maybe the metabolomic side, you could identify which bacteria can perform the same functions in the human gut.
Yeah,
because they appear to do the same thing across so many people.
Now, there was a study that did something like that. I cannot remember the author, but they studied the co-abundance groups of bacteriums. There are some certain microbes that are usually co-abundant. That means that if one bacteria has high abundance, it is likely that the second bacteria from this group will be abundant as well. So there are some groups of bacteria that behave the same in communities, and usually you can divide the whole microbiome of a person into a set of these co-abundance groups, and completely replace the species information with the information aggregated within these groups. So it is another approach we're looking forward to using co-abundance group analysis.
Very good. So what's the best way for people to learn more about what you're doing and follow it and get in contact?
Well, first of all, we have recently released the tool, this microbiomic aging clock. It is absolutely free to the public. You can reach it by typing aging.ai in your browser. Then you get to our aging clock site, where you can use blood biochemistry aging clock. And now you can also use the flora clock. We call it flora clock, the microbiomic aging clock. You can run some example data provided by us to see how it works, or you can upload your own data and download the age predictions. It also has some built-in functions like telling what probiotics will make your samples look younger or older. So we're already working in the direction of probiotic development.
Well, very good. Well, Fedor, yeah, I mean, you have a lot of work ahead of you. It's super interesting, and I really appreciate you coming on the podcast.
Well, yeah, thank you for calling me about my work. It was a pleasure discussing with you, and yes, sorry that I was a bit nervous talking with you, and sometimes it was hard to understand what I was talking about.
It's okay, no problem.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.